Hello! I hope you guys are doing all right. How are things going? I am recording and I hope everything's going all right. I see Lemuel, I see Susie, I see Jura, Marie. Good morning, Anna. Good morning, Valeria. Lemuel, always good to see you. And there's Nina. Good. I hope you guys are doing all right. Um, I guess you guys can hear me, which is important. And your quiz three. Sorry, I'm thinking of something else. Your quiz one there today. It'll open up there this afternoon, and you guys will have a week to do it. The It's basically I'll tell you what I've done here, and I'm recording this for you guys as well as my afternoon class there. The What I've done there is I've made two tests. Okay, well, I made two groups of questions, so like basically 150 questions and 150 questions there, and you know what I mean. They are what they are. And for the quiz there today, I was thinking there. I think it's open until next Sunday at midnight. So you have like 8 days to do this. I think there's 50 some odd questions in there, and I was thinking there that like a 20 question quiz or 30 question quiz. You know what I mean? There actually is a lot of material. And it wouldn't do this, and I'm not really, I don't like tests there that, you've got 1 minute a question and there's all this stress and these kinds of things. So, what I decided to do was to kind of give you like a 50 mark quiz here, but you have so much time and don't take it lightly. You know what I mean? There are so many questions that are easy peasy like this, right? I want those questions in there. But then there are some there that you'll have to dig. And the only resource there that I'm providing you there with is the PowerPoints. And some of the questions there you have to dig. But I don't expect any issues at all. The test is on Blackboard. It's all multiple choice. You do one question, another question, another question. That's all it is. And the test, I don't know if you guys have ever done, because I don't really know where you guys are at as far as your functionality regarding Blackboard. Because if you've never seen Blackboard before, it can be hideous. So it's all right. I want you guys to get used to Blackboard if you've never used it there before. So the test will open up at around 3 o'clock there this afternoon. I'll send an announcement out. And this is your quiz one, right? So please be clear. This quiz is basically an exact reflection of what your test one will be. Okay, so there's no difference between quiz one and test one in the material that they are covering. Okay, and we're going to do week 4 now. All right, which will be bacterial control. It's actually quite lengthy, so grab a chair, get some coffee going. There'll be at least 2 or 3 parts to this that will break up, and but there's viruses, prions. We get into a bit of mushrooms. We get into bacterial control. There's lots of stuff in here there for today. So my recommendation, and what do I know? But my recommendation is thank you guys for coming. Thank you guys for watching this video. This is the last video that you will be responsible for quiz 1 and test 1. Okay, so quiz 1 will open up there this afternoon. My recommendation is you're here, you're watching the video. Have some fun with week 4 because you guys have all looked over week 1, week 2, and week 3. Week 4 is just a continuation. We're not reinventing the wheel here. We're just kind of progressing along. And once you feel comfortable with week 4 material in a couple days, you look at it and you review it and this and that, make some notes from it. That's fine. And Jonathan, I see that question. I'll get there in 2 shakes of a lamb's tail. And so once you've the quiz will open at 3 there this afternoon, Jonathan. It will. But my recommendation is do not even attempt it until you're okay with week 4 material. So that's why I've left quiz 1 open. It opens there today. So at 3 o'clock, you could start it and finish it. I think there's 58 questions or something like that. You could bang them off. And you know what? If you've already done microbiology, you know, if you're taking this course as a refresher, bang, you'll be able to do the test in, half an hour. But the quiz will open at 3, and it will remain open until next Sunday at midnight. Okay, you can start the test. You can go back and look at questions. They are mixed. All right, so this is VR and virtual reality. It is open book. You guys have Google. You have your PowerPoints. You guys have everything. You guys even have each other. Aww. And I want you guys to have each other. And I hope there that y'all have met someone from the course and, talk to each other. Hey, man, this is VR. There's no rules here anymore. And giving you guys a 40-minute quiz where I have, 50 screens on and I'm watching you. No. This is how it's going to go. The quiz will be open for a week. You can start it. You can go back, look at these questions, change answers there. Next Thursday, you can do whatever you guys want. Okay? And it will be located in the assignments folder there, and I'll send an announcement out so you guys won't miss anything. And it's all multiple choice. And this one here, as I said, there's a lot of questions there that are easy peasy. Okay? And but there are some there you'll have to dig. Okay? And you'll see there today's lecture is, I don't know, it's 80 slides. Oh yeah, it's a good one. Yep. And how the quiz will be an exact reflection of what test 1 will be week 6. Now remember, this is week 4. We're going to do week 4 PowerPoints there in a little bit. And then quiz 1 will open there this afternoon. It will remain open for 8 days. It'll close next Sunday at midnight. Okay? All your answers got to be entered into Blackboard, right? So lots of time. It should be okay. And then next week there we have Thanksgiving. So there is no class there next week. All right? When I see you week 6, and don't think I'm going to be out of touch. I'm not flying anywhere. I'm right here. If there's any issues, email me. I'm not going anywhere, and I got 2 kids in school, so and my missus there, she's working she's working. So we're not going anywhere. So it's not like I'm not going to see you for 2 weeks and I'm leaving you guys all alone and all this. So if there's any issues, any questions, anything there that is irritating you, then you guys just let me know, okay? The test 1, okay, I've decided there because I gave you guys this space there to dig into research, okay, for your quiz 1. And you're not going to have to do research, crazy stuff. You're just going to go through the PowerPoints, and all questions there are directly from the PowerPoints, but you guys have Google, you guys have anything there you guys want, okay? But everything is contained in the PowerPoints. Your test 1, that will be within your lecture block. Okay, so for my morning group there, your test 1, there is no lecture there week 6. All right, that will be your test. And your test will look exactly like your quiz. But because now you've had 8 days to research and to go through your PowerPoints and and that's what I want you guys to do. I want you guys to basically to dissect the PowerPoints, okay, and know them, so that test 1, you guys will be able to do, a 70 question. Oh yeah, I'm not going to give you guys 200 questions there. I'll probably give you about 70 questions there for the test 1, but that will be in a shorter amount of time. That will encapsulate only your class. So my morning group there, your test week 6 will be from 9 to 12. Okay. The afternoon group there, your test will be from 1 to 4. Okay. And that's how it'll work. And so quiz 1, you want to do great on test 1, rock quiz 1, because they're going to look very, very similar. Different questions, mind you, same themes. That's what we're looking at. Now I see some questions over inside. So Jonathan, I hope that's okay. So the quiz 1 will open at 3, but you're going to have 8 days to do it. So there's no rush. Okay, there's no rush at all. You guys can do answer the questions there whenever you guys get a chance. Because the uncertainty of life right now, it's honestly, I'm 48. I've never seen things so chaotic. Everything, even a simple phone call to your municipality, whether it's the city of Vaughan or the city of Orangeville or, anything like this, even a simple phone call takes an hour because there's nobody there. So, and I've got single dads, single moms, I've got people working 3 shifts. So, you know what? I am absolutely accommodatable. Whatever you guys need, we'll get it done. The only thing though, be careful, test 1, week 6, it will fall within that class time. Okay? Got it? All right. I'm going through. I see Monica here on the syllabus. No book reference pages were included. Would the week 4 material be based on the... Oh, yeah. Monica, thanks for the question. The only resource that I'm asking you guys to know is the PowerPoints. Okay? The book there, accompanying book there, I think it's more for the other course there, but for this course here, from my side of it there, just the PowerPoints. You guys will see what I mean when you guys do the quiz there. Everything will be all right. Hopefully I answered that, Monica, and if I didn't, then please rephrase it. All right. Ellen, I got this. I thought the quiz is weeks 1 and 2 material and test is weeks 1 to 4. Is it? No, the test should be weeks 1 to 4. And if there's any issues, we'll let you know. Okay. That's what we're looking for there. Hopefully there. Thanks, Monica. It's great. So if there's any issues with that, you guys just let me know and we'll get it all done. I hope you guys are doing well. And I don't want this course, wherever your dreams and hopes are, this course here, I told you guys it shouldn't be an obstacle. Okay. And a pass is 70 in the course, so you guys are going to do just fine. Okay. And I'm really gunning for you guys. I'm proud of you guys for sticking this out and for upgrading your skills in the middle of a pandemic. I think it's wonderful, and I don't really anticipate any issues. Do we find quiz 1 on assignments? Hi, Jonathan. Yeah. I'll open quiz 1 there this afternoon, so you can't see it right now. And this is what we're looking for there. So if you guys keep asking questions, I love it. And people have been emailing me. Email me. I set up a WebEx there last week there because someone had some questions there. So, and please don't be afraid to get in touch with me. Okay? I'm here for you guys. If you guys need anything, away we go. Okay? If you guys have any questions, let me know. I'm going to get started because this is going to be a bit of a long one here. But the more we get through it here, it's not too bad. Not too bad. How we doing? We okay? All right. Just let me get things organized here. And we will go.
Good morning, teacher.
Hello.
Hello. I have a question. Once I didn't understand very well. Once we start the test, do we have time to finish it?
Sorry?
Once we start our test.
Oh, I know. Yeah. That's I know what you're talking about, Sandra, and that's a good question. The quiz there, like if you guys have done a Blackboard, some teachers it's called forced completion, where once you start a test on Blackboard, you got to finish it. No, this one here is not force completion. It's more of an assignment that you will enter your answers in on Blackboard. And great question, Sandra. Thank you, thank you, thank you, thank you. You can stop, go to sleep, come back to this there the next day. You see how it goes? And that's how this, it's not a forced completion. You can go back to it. You can go back and look at answers there that you did there yesterday. You can, it's completely free. Okay? And kids. But oh yeah, I got two kids at home, so we'll see what kind of disturbances they cause there this week. But Sandra, hopefully there does that answer your question?
Yes, thank you.
Okay, you're completely free. You don't have to finish it all at one shot because there are 50, I think there's 59, there may be 58 or somewhere in there. But, and I know it seems like a lot, but when you see it, you'll be banging these things off. Bang, bang, bang. There might be a couple there that give you a bit of a curveball, but I think you guys will be just fine. Okay. And good, good, good. If you have any more questions, please let me know. Other than that, let's get rocking and rolling, shall we? Okay. So here we've got, I'm going to break this up into a couple of different PowerPoints here, okay? I want to kind of continue with bacteria because that's where we left off there last week, right? But then we're going to finish off with bacteria, and then we're going to get into a little bit of virus, a little bit of mushrooms, and a little bit of I don't have my coffee here. I feel silly, but yeah, we'll get in there. Okay, mold and all these kinds of things. So let's go. Growth of microbes. Okay, is the increase in number of cells, not cell size. So the definition there of growth of microbes, and you're gonna see there that. Excuse me for a second. There we go. Thank you for your patience. Kids, eh? Oh yeah, they're gonna go. Uh, so the growth of microbes. Here we're talking, and you can have one bacteria, and the lifespan of a bacteria is about 20 minutes. So in 20 minutes you'll have two, and then four, and then 8, 16, 32, 64, 128, 256, 512. Okay, that's how these things go, right? Where like the whole idea of microbial growth is the number, okay, of cells, not the cell size. Okay, so, and one cell becomes colonies of millions. That's how it goes. That's why these guys here are such effective survivors. Okay. The control of growth is important, of course, for infection control, growth of industrial and biotech organisms. Biotech, I teach biotech, and this is biotech, what we're doing here. Basically, it's controlling microorganisms, and this is the future. You look at the future here. It's in Canada didn't have a vaccine production facility. That's why we were behind the 8 ball pretty much the entire pandemic, but these things will never happen again. Every country in the world will have its own vaccine producing factory. So it's a good time to get into the biz, I tell you. And they will need MDR, medical device reprocessing, for sure. There are 3 kinds of bacterial growth. Okay, for bacteria, okay, it's what we call binary fission. And then these 2 guys here, these are for your mushrooms, your yeast, your molds, these kinds of things. And we'll get into these, all right? So for bacteria there, it's called binary fission. The idea of binary fission comes from when an expanding cell divides, it becomes 2. And just basically, I'm very visual myself, all right? And you'll see here there that there's your bacterium there, DNA nicely tightly coiled. Okay, and I had a question there last week from a student and it was a good question. Like, this is a microbiology course in a sense, but you don't need the microbiology to get a great mark in it. It will help in your facilitation of understanding, but I've organized a test there so you don't need a heavy duty background in science. Okay, you just need to be aware and be able to, if you've read over the PowerPoints, you'll be just fine there for the quiz and the test. Okay, and you'll see here that number 2 there, the DNA of the bacterium is replicated, and you see there's no difference there to the structure of the cell wall. Okay, you just got 2 sets of DNA in one bacteria. And then you'll see there the DNA starts to get pulled to either side, kind of like the North Pole and the South Pole here. And then you'll see that it starts to separate, okay, more so, and then you get the cell wall that actually goes in here, and that's what we're talking about. This is called binary fission. Okay, so you end up with one daughter cell, all right, and they are an exact replicate of the first one. And just to go back here, the binary fission occurs when an expanding cell divides and becomes 2. Depending on the species of bacteria, this can happen in many ways. It can happen longitudinally, transverse, symmetrically, blah, blah, blah. That's, what I'm asking you to know is that bacteria divide, reproduce through binary fission. Okay. Most common form of cell division among prokaryotes, right? And you guys will remember there that a eukaryote is us, lizards, whales, trees, mushrooms. Prokaryotes are the bacteria, right? They're a simpler form of, they were the first, in theory, that's what we think, to develop here on Earth. Okay? And you know what I mean? These are just some pictures here of this whole process here of binary fission. Okay? Just to tell you that I'm not lying to you. Okay. Here we get into, this is more common in yeast, and this is what we call budding. And you guys can see over here of where we're getting at. Okay. And budding is, like when you think of budding, think of yeast. Did I just say that? Budding begins when a small protrusion expands outward from a mother cell, as you can see right here. Okay. This protrusion will enlarge, okay, and eventually forming a new daughter cell. Okay. One of the issues, though, is you see the genetic material in here, it's got to make its way into this bud. And if it doesn't, then you've got an incomplete bud. You've got an incomplete cell that'll probably just get whacked and it'll get destroyed. Okay, so the newly replicated DNA must make its way into the protrusion for the process to work properly. All right, and it's not common in bacteria, but yeast. And I got some good pictures there of yeast and all this kind of stuff right here. So here's just another shot of this budding, and this is a real picture of what's happening. So there you go. There's your budding. So the other kind of microbial growth there, dealing with fungi, is what we call filamentous growth, mycelial, and it's these tentacles. That's why I'm going like this with my hands. I'm not saying hi to you, but hi to you guys anyway. It's these tentacles that grow under the ground. Like when you guys see a mushroom, like a mushroom cap. Do I have a picture of one? I think I do somewhere down below. But if you guys see a mushroom cap, that's the mushroom there that we eat. Okay? That's the end game of a mushroom. The mushroom is actually down in the ground, these filamentous, these mycelial growth, these like spider webs, but it's not, okay? All in the ground here. And they're all, kind of independent, but they all come together to make this mushroom. We'll get in there. But filamentous growth, some microbes grow in a linear fashion, Okay, typically in response to some form of chemical stimuli, involves lengthening of its tube shapes. And we'll show you these. You guys can see down here what's going on. Multinucleated cells are usually created. Multinucleated, okay? The nucleus that houses there the DNA. You can have multinucleated cells, okay? At certain lengths, the tube will branch and expand in different directions. It'll just go. That's that kind of spider web nature that these filamentous growth mushrooms there they have in the ground. Okay, very common in fungi, as I just said, and you'll see there it starts off with a spore. Okay, there's a germination. All right, and then you'll see how it just kind of creeps along and grows another extension and another extension and another extension. Right, it's just creeping. Okay, it's these filaments. And you'll see what goes on here. It creates these septa there that divide the cells there. And you can have multinucleated this and that. Okay. And good word to know, hyphae. These are these filamentous growth, these extensions. We'll get more into those. And here's just a picture, okay, of this filamentous growth. And you'll see there it's segmented. And you can actually see the extensions growing. And and just to let you know there that there are names here of these bacteria and all these kinds of things here. I do ask for 2 or 3 exact names of a bacteria or the yeast or whatever that I'm asking for there in the quiz, and I will in the test. But I'm going for microorganisms that are crazy important that you should know. Okay, and these kinds of things. So, but again, it's open book. Everything is there. There's no surprises. If you just dig through the PowerPoints, you'll find every answer and get 100%. That's how she goes. Okay. Regulating growth. Yeah, there's a couple of them here. There's the nutrients. And like when I worked in a mushroom farm, up by in Gray County there a million years ago, what we would do is you'd get a plastic bag, heavy duty plastic bag. You'd fill it with some type of substrate, cornmeal, oats, whatever this particular mushroom was after. And yeah, you'd put about a kilogram in or a pound. I think it was a pound of corn millet or oats or whatever. You'd add the correct amount of water because you need that proper humidity, right? That just might be a test question. What's the proper humidity? But and then you would add nutrients like phosphates. You'd add calcium and magnesium. Everything needs nutrients. Everything needs vitamins, right? The whole idea there that why do we eat things? Because we can't make them ourselves, right? So nutrients, they regulate growth. Huge. Environmental conditions, absolutely regiment growth. Okay, temperature, crazy important. pH, okay. We did a little bit of pH there last week. pH is a measurement of acids, okay. And we went into that there last week. An acid is anything under a pH of 7. A base is anything over a pH of 7. And you see distilled water is a pH of 7. Okay? Our blood is a pH of about 7.4. And I mean 7.4. And not 7.8 because that would be trouble. And so the pH is just it's just a measurement scale there that we measure acids and bases in. And everything has to be in tune. Okay, so pH definitely. Okay, I think the microorganisms there, they like pH. They're somewhere between 6.5 and 7.5 pH. Okay, osmotic pressure, osmosis, the movement of water across membranes. Absolutely, you got to have enough water in your cells there to move things around. And generation time, absolutely. How fast do these things reproduce? Okay, so those are factors there affecting growth. We've got these ones here. Oxygen, You'll remember there that bugs, and I mean bugs, not six-legged, I mean microorganisms, bugs there, if they like oxygen, they're aerobic. If they don't like oxygen, they're anaerobic, means without oxygen. There's your pH, osmotic pressure. Oh yeah, UV radiation. Okay, you'll remember ionizing radiation, atmospheric pressure, all these kinds of things. They are growth factors. Okay. Optimal microbial growth ranges. Every microorganism has an optimal range. Okay, I'm not going to ask you there for specifics of this. I'm more in tune of generalities. Since bacteria have colonized every possible environment niche on the planet, they have adapted to every possible environmental pressure. Yeah, they're I think it was a Russian expedition there. They were in, I think it was just last year, and they were in Antarctica and they were drilling. They were taking ice cores samples. They were drilling, they could see there, I think it was a kilometer or two below the ice, they could see a freshwater lake had formed underneath the ice. And, they did some core samples. They drill the ice and they take this ice up and they can tell how much oxygen was in the air 100,000 years ago, these kinds of things. But the bacteria, they found some. So there we go. We've done all that there. The microbial temperature, there is an optimal for each one. And you'll see there that there is a bit of space, okay? They do like a certain temperature, all right? But then, as with any heat, it becomes a problem after a while. And if you throw an egg into a hot frying pan, right? What happens to the egg? An egg is 100% protein. Well, I shouldn't say 100%, but you know what I mean. An egg is solid protein, and when you throw that egg into a hot skillet, what happens is now the egg starts to turn white, hard, and solid, right? Even the yolk as well, because what the heat is doing, it's denaturing the proteins. These proteins that your body makes, right? And remember we covered that? The mRNA comes out of the outside of the nucleus, goes to a ribosome. Ribosomes are protein makers. You know that one too. And these proteins get made one amino acid at a time. And the longer they get, what'll happen is they'll start to, and you have to think three-dimensionally. These proteins will start to twist and turn and create these amazing and quite elaborate three-dimensional models. And the reason why these proteins make this exact three-dimensional model, because they have a function. All right? And function, anatomy and physiology is function tied to structure. That's what it is, right? So everything, things have a structure because the structure is tied to the function. Look at my hands. They're like this because they have a function. This is their structure, and the structure is directly related to their function. And all these proteins there, they become denatured, okay, once the heat starts to go up. And you'll see there that optimal microbial growth starts to go down quite quickly. So you see, what have we got here? We've got psychrophiles, we've got psychotrophs, we've got mesophiles, we've got thermophiles, and hyperthermophiles. These are different bacteria. This is a good little slide here. I would have a look at this because there's a couple of things here that I'm not going crazy into these, okay? These are just some words there of kinds of bacteria. So, okay, so we got psychrophiles. They grow well at around 0 degrees. Okay? Optimal up to about 15. These are found in polar regions or at the bottom of the ocean. And you have to think, the top of the ocean will be 28 degrees. Beautiful, sunny. I'm swimming in it. We're all swimming in it. It's great. But as you go down to the bottom of the ocean, oh yeah, it gets freezing. freezing down at the bottom of the ocean, unless you're by some type of lava vent on the bottom of the ocean. But that's these guys here. Here we've got psychotrophs, where their optimal growth is between 20 to 30 degrees. Major factors in spoilage of refrigerated foods. Okay? So, like botulism, these ones right here. Mesophiles, optimal growth between 20 to 45. Most microorganisms, they fall into this category. Okay? Therm, okay? Thermometer means heat. So you have a look at these two here. Don't do that, Ian. Thermophile, and we got hyperthermophiles. Hyper means up. Hypo means low. So if you had a hypothermophile, it might be lower. So we got thermophiles here. They tend to grow better in hotter temperatures. Okay? Hot water lines, hot springs, these kinds of things. And then you actually have hyperthermophiles. These ones here, they're oddballs. Okay? At the area there on the seafloor, they'll live by thermal vents. Okay? So you'll see here there that we've got some optimal temperatures there. Y'all can see these here. There's your psychrophiles, your psychotrophs, your mesophiles. And you'll see here the majority of microorganisms are these guys right here. Okay? Because these ones here tend to be extremes. Not normal. Okay? Bugs that are living at 90 degrees, that would tear most cells apart. Okay? In each case there, at the minimum temperature, the plasma membranes begin to solidify and the enzyme function slows. Okay? So you think there that the colder something gets, the more things start to shut down. And we've, if you haven't heard of these stories there, like in the wintertime, you know, people will take their snowmobiles and go across the ice, you know, in the bay. And usually come around Christmas time or January there, the ice is like this thick. You can drive a Ford F-150 truck across it. Okay, fine. But sometimes there, in a thawing part there, the ice will get thin, and you hear, not all the time, but you hear sometimes there about people going through the ice. And, it's a scary situation. The threat of death is very high. But what'll happen is you've heard about it there, a child will go underneath the water for 20 minutes. And normally there's no chance of survival. But what happens is in the very cold water, organ systems start to not shut down, but go dormant. All right, and arms and legs will, that's why they say don't go swimming after a big meal, because all the blood rushes to your stomach. You don't have any blood in your, you have minimal blood in your arms and legs. So if you get into an emergency situation out in the water, your arms and legs really won't save you because they don't have enough blood because you're too busy digesting lunch. So the whole thing here is when the temperature gets colder, things start to slow down. Okay, that's why the optimal microbial temperature for like mesophiles isn't 5 degrees, right? Because it's too cold for things to move around. Okay, maximum temperatures there, the plasma membrane becomes too fluid. Yeah, and at higher temperatures there, the proteins will start to denature and things will just start to blow apart. Okay. Oxygen level concentrations need to be tightly regulated for most microorganisms. Aerobic respiration with oxygen serves as the terminal electron acceptor. Okay, this will just make absolutely no sense if you're not a science person, and you don't need to know it there for the quiz or the test. All you'll need to know, oxygen is for aerobic respiration. Okay. For non-aerobic respiration, microbes employ fermentation for the creation of energy. There's even some there that are anaerobic without oxygen. You get these thermophiles there that live at the bottom of the ocean. They don't breathe oxygen. They breathe sulfur, these kinds of things. They're just hideous, poisonous to us, but that's what they do. Okay. There are 4 types of oxygen classifications for microorganisms. Okay, this is a good little slide here. Sorry, I had a twitch here. I'm gonna have to pause this, and I apologize. I've gotta. Actually, no. I'm gonna finish this. No, I'm gonna pause it. I apologize. I've gotta pause it. So I'm gonna mute it. I'm gonna pause it. Okay, and I'll have to start a new video. Sorry, guys. I'm going to get us out of this. Nothing I can do. I got kids at home, and this is it. But I'll be back in 2 seconds. I'm gonna pause this and stop the video and stop the recording. I'll be back.